Welcome back to Module 1 of Cells, Tissues and Regulation. Topic 1.2 will be an overview on cell division, cell replication and protein synthesis. During the course of this video, try and answer as many questions as you can from Module 1, Topic 1.2. The learning objectives for this topic are to describe the differences between the structure and function of the DNA and RNA, describe the role of RNA in the production of proteins, describe the process of mitosis, and describe the process of DNA replication. All the known life forms on planet Earth rely on large biomolecules called nucleic acids, which are absolutely essential for life. Nucleic acids include two major classes of molecules, DNA and RNA. These are the largest molecules in our body. Nucleic acids are composed of carbon, oxygen, hydrogen, nitrogen, and phosphorus groups. The structural unit of a nucleic acid is called a nucleotide, which are quite complex. Each nucleotide consists of three components: a nitrogen-containing base, a pentose sugar, and a phosphate group. There are 5 different nucleotides, depending on the variety of the nitrogen-containing base present. DNA and RNA are made from these nucleotides. Now let's learn about them individually. DNA is a long double chain of nucleotides that are coiled into a spiral staircase-like structure called the double helix. It is located inside the nucleus in the form of chromatin. DNA is made up of 4 different nucleotides: adenine, thymine, guanine, and cytosine. The pentose sugar in DNA is called deoxyribose, as reflected in the name. The bonding between these nucleotides is very specific. An adenine, or A, will always bind with a thymine, or T, while guanine, or G, will always bind with cytosine, or C. And because of this specific binding between the two nucleotides, they are called complementary bases. The 2 complementary nucleotides are linked by hydrogen bonds, as seen in the figure. DNA holds all the genetic information in our body and directs protein synthesis. Proteins are essential for growth, repair, and functioning of all living cells. Life cannot be sustained without proteins, including enzymes, hormones, antibodies, and muscle proteins. DNA must first replicate before cell division, and the genetic material it holds is then passed on to the daughter cells after division. The RNA, on the other hand, is a single strand of nucleotides that is mainly located outside the nucleus of the cell, in the cytoplasm. RNA is also made up of 4 different nucleotides, and they are adenine, thymine, cytosine, and uracil, which replaces the thymine found in DNA. The function of RNA is to carry the DNA's instructions for protein synthesis to the ribosomes. RNA is further classified into 3 subtypes, and they are messenger RNA, transfer RNA, and ribosomal RNA. The 3 types of RNA carry out very specific functions in the process of protein synthesis. The messenger RNA, or mRNA, carries instructions from DNA to the ribosomes for building polypeptides. Transfer RNA, or tRNA, transports the amino acids to the ribosomes, where they decode the mRNA to build polypeptides. Finally, ribosomal RNA, or rRNA, along with other proteins, build ribosomes, and also assist in linking the tRNA to amino acids. As mentioned earlier, the DNA in the nucleus is present in the form of chromatin, and when we look at the structure of chromatin, it looks like a granular thread. Chromatin is typically made up of 30% DNA, 60% histone proteins, which tightly package and regulate the DNA, and 10% of newly formed DNA. The fundamental units of chromatin are called nucleosomes, which consist of flat disc-shaped clusters of 8 histone proteins that are connected like beads on a string by the DNA molecule. Chromatin plays a key role during cell division as it can tightly coil and condense to form chromosomes when the cell is preparing to divide. Now let's learn a little bit more about histones. Histones provide the physical means for packing the very long DNA molecule into a compact and orderly fashion. Furthermore, they also play an important role in gene regulation as they have the ability to activate or shut down the DNA molecule. For example, when the cell is not dividing, the presence of methyl groups on histone proteins can shut down the nearby DNA, whereas when a cell is ready to divide, the presence of an acetyl group on the histone proteins can activate the nearby DNA. Our body is made up of trillions of cells, many of which need to divide constantly for various reasons such as growth and tissue repair. The exception are neurons or nerve cells, which lose their ability to divide when they mature. Cell division is a process where a parent cell is divided into two new daughter cells. There are two distinct types of cell division. In mitosis, the chromosomes in the nucleus of a parent cell are duplicated and given to two new daughter cells. All the cells in our body, other than the sex cells, or gametes, undergo mitosis. In meiosis, the number of chromosomes in the daughter cells are halved, which means that all the daughter cells would have half the number of chromosomes of the parent cell. Meiosis is seen in gametes. Gametes are the sex cells, such as the sperm and eggs. The cell cycle is a series of changes that a cell undergoes from the time it is formed until it reproduces or divides. Whenever a new cell is formed, it automatically enters into interphase, where the cell grows and carries on its usual activities. The interphase is further divided into three subphases. In the G1 phase, the cell becomes metabolically active by synthesizing proteins rapidly and growing vigorously. In the second phase, or the S phase, the DNA is replicated, so that the two new daughter cells will receive identical copies of DNA once the cell divides. At the same time, new histones are being made and assembled with DNA to form chromatin. In the final phase, or the G2 phase, the enzymes and other proteins needed for cell division are synthesized and moved to their respective sites. By the end of the G2 phase, the cell completes replication of its centrioles and DNA. During each of these subphases, the cell continues to grow and carry out its usual functions. After interphase, the cell starts the mitotic phase, which is again divided into four different subphases. We will learn about cell division later in this video. Before a cell can divide, its DNA must be replicated into two identical copies. The cell can then pass one copy to each of its daughter cells. The replication of the DNA begins in the S phase of interphase. The process of DNA replication involves two major enzymes. Helicase is an enzyme that unwinds and separates the DNA helix into two DNA strands by breaking the hydrogen bonds between the complementary bases. The area at which the hydrogen bonds are broken between the complementary bases, which is where the DNA strands separate, is known as the replication fork. The separated strands of the DNA molecule act as a template to which the enzyme DNA polymerase adds the complementary free nucleotides along the template strand, forming two new DNA strands. Because the DNA polymerases work only in one direction, the two new strands, called the leading strand and the lagging strand, are always synthesized in opposite directions. Once the DNA polymerase enzyme has attached the free nucleotides, the enzyme ligase connects short segments of DNA together, restoring its double helix structure. In terms of DNA replication, there are a few things that you should remember. The enzyme helicase unwinds the double helix DNA molecule and exposes its bases. DNA polymerase attaches free nucleotides along the template strand. Two new strands are synthesized in opposite directions, called the leading strand and the lagging strand, because the DNA polymerase enzyme only works in one direction. Finally, once the free nucleotides are attached to the strands, the enzyme ligase splices or joins the DNA fragments, restoring its double helix shape again. Once DNA replication is completed, the cell then moves into the process of cell division, where the replicated DNA is distributed to two new daughter cells. This is called mitosis. Mitosis is divided into 4 phases, with each phase merging smoothly into the next one. The first phase is prophase, which consists of early prophase and late prophase. In early prophase, the DNA, which is in the form of chromatin inside the nucleus, tightly coils and condenses to form chromosomes. At the same time, the microtubule assembly, called the mitotic spindle, forms, with centrosomes acting as their focal points. In late prophase, the nuclear membrane surrounding the chromosomes slowly breaks down, allowing the spindles to interact with the chromosomes. In metaphase, the centrosomes are at the opposite ends of the cell, while the chromosome cluster moves to the center of the cell, with their centromeres aligned at the spindle equator, which is also known as the metaphase plate. In anaphase, the chromosomes split, and a complete set of chromosomes are pulled toward each end of the cell. Telophase, which is the final phase of mitosis, begins when identical sets of chromosomes reach the opposite ends of the cell. Here, the chromosomes then start to uncoil and resume their thread-like chromatin structure. A nuclear envelope forms around each chromatin mass, and nucleoli reappear within these new nuclei. The mitotic spindle breaks down and disappears, thereby ending mitosis. For a brief period of time in the cell, it is binucleate, meaning that it contains 2 nuclei. Cytokinesis, which is the division of the cytoplasm, begins during late anaphase and continues through and beyond telophase. Once the cell division is complete, the cell starts the cell cycle by entering into interphase again. Now let's move on to protein synthesis. Proteins contain long chains of amino acids that play a crucial role in sustaining life. Much of the metabolic machinery, both in our body and the cell itself, are related in some way with protein synthesis. For example, muscles which are used for movement are composed of protein fibers. The immunoglobulins, antibodies, and enzymes that perform complex physiological functions are also proteins. A protein is a long folded chain of amino acids. The process of protein synthesis is controlled by DNA, which determines the nature and position of an amino acid in a protein, thereby determining its amino acid sequence and therefore its function. A sequence of 3 nucleotide bases in the DNA are called a triplet and codes for a specific amino acid. The sequence of triplets in each gene gives precise instructions on how a particular polypeptide is to be made. It specifies the number, type, and order of amino acids needed to build a particular polypeptide. Variations in the arrangement of nucleotides allow our cells to make different kinds of proteins. Genetic mutation is a change in the DNA base sequence that may lead to incorporation of incorrect amino acids in particular positions of a protein. The affected protein in some cases can lead to medical conditions, such as cancer. Now let's briefly look at how the protein is synthesized from DNA. Whenever there is a need to synthesize a particular protein, the DNA inside the nucleus needs to be decoded into mRNA and sent to the ribosomes for the synthesis of proteins. The process by which the DNA's information is encoded into mRNA is called transcription. Once the mRNA is produced, it then leaves the nucleus through nuclear pores and enters the cytoplasm. The information carried by the mRNA is then decoded in the ribosomes, where it is used to assemble amino acids into polypeptide chains. This process is called translation. So we'll just go back to transcription for a moment. Transcription is a process where the DNA's information is encoded in mRNA. Transcription cannot begin until gene-activating chemicals, called transcription factors, stimulate histones at the gene transcription site. The transcription factors then bind to the promoter region, which is a special DNA sequence that contains the start point of the gene to be transcribed. Furthermore, it also helps in binding the RNA polymerase enzyme to that promoter region. Once these preparations are made, RNA polymerase can initiate transcription. The RNA polymerase enzyme is responsible for mRNA synthesis. It synthesizes mRNA by unwinding DNA and adding complementary free RNA nucleotides, and then joining the DNA strands back together into a double helix molecule. Transcription stops once the enzyme reaches the termination signal. Transcription involves three basic phases. In the first step, or initiation, RNA polymerase pulls apart the strands of the DNA double helix and binds to the start point in the promoter region with the help of transcription factors. The transcription factors also determine which strand is going to serve as the template strand. The DNA strand that is not used as the template strand is called the coding strand because it has the same nucleotide sequence as the mRNA that is to be built. In the second step, called elongation, RNA polymerase aligns the incoming free RNA nucleotides with the complementary DNA bases on the template strand and links them together. As RNA polymerase elongates the mRNA strand one base at a time, it unwinds the DNA helix in front and then rewinds the double helix behind it. In the final step, termination, the RNA polymerase reaches a special base sequence called the termination signal. Transcription ends here and the newly formed mRNA separates from the DNA template. Translation is a process where the genetic information carried by the mRNA molecule is decoded by ribosomes in order to make a particular kind of polypeptide. Translation involves all three types of RNA, the mRNA, tRNA, and rRNA. After mRNA synthesis, the mRNA leaves the nucleus and attaches to a small ribosomal subunit. The small ribosomal subunit scans along the mRNA until it finds the start codon. A codon is a 3-base sequence on the mRNA that complements the 3-base sequence on the DNA. The initiated tRNA anticodon recognizes the start codon on the mRNA and binds to it within the ribosome. After this, the large ribosomal subunit attaches to the small subunit and forms a functional ribosome. The anticodon, which is the 3-base sequence on the tRNA, binds to the complementary codon of the mRNA and adds its amino acid to the protein chain. The mRNA strand is read sequentially, and new amino acids are added by the tRNA until the ribosome reaches the stop codon, which signals the termination of protein synthesis. The following figure briefly illustrates the process of protein synthesis. Prior to initiating protein synthesis, the cell must synthesize mRNA in the nucleus. The newly synthesized mRNA leaves the nucleus and gets picked up by the ribosomes in the cytoplasm. At the same time, the enzyme amino-acyl tRNA synthetase attaches an amino acid to tRNA, according to its anticodon. The amino-acyl tRNA carrying a specific amino acid then diffuses to the ribosome. A ribosome consists of 3 active sites, the A site, the P site, and the E site, that each act as binding sites for the amino-acyl tRNA and mRNA. Translation begins when the initiator amino-acyl tRNA recognizes the complementary start codon on the mRNA and binds to it at the P site, leaving the A site vacant and ready for the next amino-acyl tRNA to deliver its amino acid. The incoming amino-acyl tRNA binds to a complementary codon in the site A of a ribosome. An enzymatic component in the ribosome binds the amino acid of the tRNA in the A site to the amino acid of the tRNA in the P site. As the ribosome translocates or moves, it shifts the codons along the mRNA. This shift moves the tRNA from the A site to the P site. The unloaded tRNA is then transferred to the E site from where it's released. only to be bound or recharged by another amino acid in the cytoplasm. This conveyor belt-like process of attaching amino acids to the polypeptide continues until the stop codon is read at the A site. Once the ribosome reaches the stop codon, it triggers the ribosomal subunits to separate, thus releasing the mRNA and the new polypeptide. A polyribosome is a complex of one mRNA being read by several ribosomes simultaneously. Polyribosome arrays allow for a single strand of mRNA to be translated into hundreds of the same polypeptide molecule in a short time. As the ribosomes read the mRNA from left to right, new ribosomes are added on the left-hand side at the start of the RNA. And once the ribosomes have reached the stop codon of the mRNA, the subunits then dissociate from the mRNA. As described earlier, ribosomes in a cell can attach and detach with the rough endoplasmic reticulum. However, if a protein that is being synthesized has an endoplasmic reticulum signal sequence, the ribosomes, along with its mRNA, attach to the rough endoplasmic reticulum. The signal recognition particle guides the ribosomes and the mRNA to an appropriate receptor site on the endoplasmic reticulum. Once the proteins have been synthesized, they are folded inside the endoplasmic reticulum. In some cases, molecules such as sugar groups may be added to the protein and sent off to the Golgi apparatus in a transport vesicle, where the protein is processed further. This slide shows how the information is transferred from the DNA of the gene to the complementary mRNA molecule, whose codons are then translated by tRNA anticodons. The triplets on the DNA sequence code for a particular amino acid in the polypeptide. The 3-base sequence, or codon, of the transcribed mRNA complements the triplet on the DNA. Consecutive bases of tRNA anticodons recognize and bind to the mRNA codons, during which amino acids are added to the polypeptide in ribosomes. If you notice carefully, the tRNA anticodon resembles the triplet sequence of the original DNA genetic code, except that the T is replaced with U. From this video, we have learnt about the differences in the structure and function of DNA and RNA, the different types of RNA and their functions, how DNA is replicated and distributed during cell division, how RNA is synthesized through transcription, and finally, how proteins are synthesized.